Mitosis. Mitosis is used for creating identical copies of cells for uses such as repairing the body and is only used for replicating somatic cells. There is no variation in mitosis unless there is a random mutation that occurs in the replication of the DNA. When there is a mutation, the cell's function could be altered, causing issues that could be potentially fatal to the organism. Mitosis is not used in sexual reproduction because there is no genetic variation which would affect the evolution of the organism. But it is used in asexual reproduction because the parent's offspring is an identical copy of itself, thus variation is not wanted. In both the drawings of metaphase and anaphase, the green chromosome is the maternal and the red paternal. The purpose of metaphase is to arrange the chromosomes in such a manner that it ensures that each cell will contain one sister chromatid from each of the replicated chromosomes. Metaphase begins after prophase when the nuclear envelope dissolves and the spindle fibers begin to elongate. During metaphase, in mitosis, the chromosomes are aligned in the center of the cell by the mitotic spindle. As this occurs, the chromosomes migrate to opposite sides of the cell, and the spindle fibers begin to elongate and attach to the chromatids. Anaphase is meant to separate the sister chromosomes to opposite sides of the cell in preparation for the splitting of the cell. Anaphase begins as the spindle fibers shorten, resulting in the sister chromosomes being pulled to opposite sides of the cell. As this occurs, some of the spindle fibers on the two sides of the cell lengthen, stretching the cell outwards in preparation for the cell to split. As this occurs, some of the spindle fibers on the two sides of the cell lengthen, stretching the cell outwards in preparation for the cell to split.
Meiosis is used in germ cells for variation in offspring. There is a huge amount of variation in this method due to random fertilization, crossing over, random mutations in the S phase of interphase, and how the triads assemble in metaphase. Meiosis is a method used in sexual reproduction in germ cells in order to ensure variety. Variety is wanted because of the Red Queen theory, to keep up with the constantly changing environments around the organism. Homologous pairs in prophase have exchanged tips or crossed over parts of the chromatid in exchange for parts of the chromatid in the other homologous pair. This ensures mixing of the DNA and more variation. During metaphase 1 of meiosis, homologous pairs are aligned by spindle fibers in the center of the cell. The orientation of chromosomes is completely random, which contributes further to variation. This random orientation is called independent assortment. Metaphase 1 is necessary because the chromosomes must be aligned and matched with their pair for when they are split into different cells. In anaphase 1 of meiosis, the homologous chromosome pairs are separated to each side of the cell by the spindle fibers. This separates the chromosomes so that the cell can divide with the right amount of DNA in its daughter cells. In metaphase 2 of meiosis, the chromosomes are aligned in the center of the cell by the spindle fibers in no particular order. The purpose is to organize the chromosomes so that they can be divided easily. In anaphase 2 of meiosis, the aligned chromosomes are split apart into non-identical chromosomes that are moved to each side of the cell. The purpose of this is to divide the chromosomes for cell replication.
The differences between mitosis and meiosis. Mitosis is the process for which replicating an exact copy of a somatic cell, while meiosis is the process for the replication of sex cells. In mitosis, there is only one metaphase and one anaphase, creating two diploid daughter cells. In meiosis, there are two metaphases and two anaphases, creating four non-identical haploid cells. In mitosis, there is almost no variation with the exception of random mutations. But on the other hand, meiosis has much more variation from crossing over and also has random mutations. These are all the differences between mitosis and meiosis.